Imagine the terror of the Green River Killer Gary Ridgway, terrorizing a community for decades, leaving behind a trail of victims. Or the baffling complexity of the John Bennett Ramsey case. What happens after DNA is meticulously collected and amplified? In the forensic lab, fragmented genetic whispers are transformed into undeniable profiles, linking suspects to crime-solving cold cases like the Golden State Killer, and even freeing the innocent, as seen in the Central Park 5 exonerations. Welcome back, future justice seekers. In our previous episodes, we explored how forensic and ancient DNA samples are collected and how challenging, degraded DNA is extracted and amplified. Now in Part 4, we step squarely into the forensic lab to understand the powerful art and science of interpreting those amplified DNA products into profiles that can secure convictions or reveal innocence. After the rigorous amplification we discussed in Part 3, our lab technicians aren't just looking at a tube of amplified DNA. They're ready to create a distinct genetic fingerprint using what we call short tandem repeats, or STRs. STRs are short sequences of DNA, typically 2 to 6 base pairs long, that are repeated multiple times in specific locations on our chromosomes. What makes them so powerful is that the number of repeats varies greatly from person to person. For example, one person might have 10 repeats of a specific STR sequence at a certain location, while another might have 12. Since we inherit one copy from each parent, we have 2 numbers for each STR locus. When forensic scientists examine about 20 specific STR loci across the human genome, as is standard in the US using the CODIS core loci, the chance of 2 unrelated individuals having the exact same combination of repeats at all those locations is astronomically small, making STRs incredibly individualizing. Once amplified, these different-sized STR fragments are separated and visualized using a technique called Capillary Electrophoresis, CE. Think of it like sorting different-sized beads through a very fine electrically charged sieve. As the DNA fragments pass through the capillary, a laser detects a fluorescent tag on each fragment. This data is then translated into an electropherogram, a digital graph showing peaks. Each peak represents an STR allele, a specific number of repeats. The height of the peak relates to the amount of DNA, and the position tells us the size of the fragment. The electropherogram is the forensic scientist's map to identity. Interpreting it correctly is crucial for solving cases and avoiding miscarriages of justice. Single-source profiles, the easiest to interpret. These come from a single individual and show 2 peaks, alleles, at most, for each STR locus, one from each parent. A clear, clean profile means a direct comparison can be made to a suspect or a database. Mixed DNA profiles, often encountered in real crime scenes where multiple individuals contributed DNA, e.g. a victim and an assailant. These profiles show more than 2 peaks at some or all STR loci, making interpretation complex. Specialized probabilistic genotyping software, like TrueAllele or STR-Mix, uses advanced algorithms and statistical models to deconvolve these complex mixtures, assigning statistical weights to potential contributors. This was vital in ongoing efforts in cases like the John Bennett Ramsey investigation, where mixed and low-quantity samples present continuous challenges and require these advanced software tools. Low copy number (LCN) profiles. When dealing with very small amounts of DNA, the electropherogram can be messy. It might show allelic dropout, where an allele is present but fails to amplify, stutter, small peaks that are artifacts of the PCR process, or contaminant DNA. These stochastic effects require careful interpretation and often generate less statistically powerful results, but can still be highly informative. The power of exclusion, perhaps even more powerful than inclusion, is exclusion. If a suspect's DNA profile does not match the profile from the crime scene, they are excluded as the source of that specific evidence. This is what happened in the groundbreaking Central Park 5 case, where DNA evidence collected years later definitively excluded the convicted men, leading to their exoneration. This highlights DNA's dual power, to convict the guilty and free the innocent. One of the greatest revolutions in forensic DNA has been the creation of searchable databases, The most famous is CODIS, the Combined DNA Index System. CODIS is a national DNA database maintained by the FBI in the United States, linking federal, state, and local forensic DNA databases. It contains DNA profiles from crime scene evidence, unknown individuals, convicted offenders, arrestee samples, missing persons, and unidentified human remains. When a forensic lab generates a DNA profile from a crime scene, it can be uploaded to CODIS and searched against millions of existing profiles. A hit means the crime scene DNA profile matches a profile already in the database, potentially identifying a suspect or linking multiple crime scenes together. CODIS has been instrumental in solving countless cold cases. For example, the Green River Killer, Gary Ridgway, who operated for decades, was finally linked to numerous murders through DNA evidence. Initial CODIS searches didn't yield a hit, but as technology improved and new samples were entered, DNA collected years earlier eventually connected him to victims, leading to his conviction. Similarly, while the ultimate identification of the Golden State Killer, Joseph DeAngelo, involved advanced genetic genealogy, which we'll discuss in Part 6, initial efforts did involve submitting cold case DNA to CODIS, illustrating the typical first step in these types of investigations. Beyond standard autosomal STRs, other specialized DNA analyses are deployed for specific forensic questions. Y-STRs, these markers are found only on the Y chromosome, meaning they are inherited directly from father to son. They are particularly useful in cases involving sexual assault with multiple male contributors, or when trying to identify a male lineage, as all males in a paternal line will share the same YSTR profile. Mitochondrial DNA, mtDNA. As we discussed in Part 3, mtDNA is present in hundreds or thousands of copies per cell, making it incredibly useful for highly degraded samples, such as old bones, hair shafts without roots, or very fragmented remains. It is inherited directly from mother to child, tracing a maternal lineage. For example, mtDNA was critical in identifying victims from the 9/11 attacks where traditional nuclear DNA was highly degraded. Finally, the DNA evidence arrives in court. Forensic DNA experts serve as expert witnesses, explaining the science to juries. The power of DNA evidence often comes down to statistical significance, expressed as a random match probability, RMP, the chance that a randomly selected person from the population would have the same DNA profile as the evidence. Numbers like 1 in a quadrillion are common, demonstrating the immense power of DNA to identify. The chain of custody, which we discussed in Part 1, is meticulously reviewed to ensure the integrity of the evidence presented. From peaks on a graph to probabilities in court, the interpretation of forensic DNA profiles has revolutionized criminal justice. It's a testament to science's ability to speak for victims, identify the perpetrators, and safeguard the innocent. Next time in Part 5, we take our genetic journey to its cutting edge, ancient genomes, and the future of paleogenetics. We'll explore how DNA to unlock unprecedented secrets from the distant past. Make sure to subscribe to The Geneticist, like this video, and ring that bell to catch all our deep dives into genetics. See you soon!